Hello all. My apologies. I had a ton of trouble with Zoom getting it to start up. Could not get the host code to work. So we are going to start this webinar 18 minutes late. But it looks like, oh, all right. Well, the room is filling up, so this is going to be okay still. Thank you all for waiting, and I apologize for such a late start to this thing. All right. I am going to share my screen. And we'll get going here. All right. So here we go. Unmixing and compensation in FlowJo software. I am going to make sure that I have the chat turned on so that I can see any questions that pop up, and I'll try to answer them in real time as we go through. I am John Quinn, the Director of Science and Product Development for BDB Bioinformatics. My email address is up there on the screen. It is john.flojo@bd.com. All right. So the plan for today's webinar is that I will briefly do the theory behind compensation and unmixing. We'll review what the current options are, and then I'll show you how to do it in FlowJo. And then at the end, well, we'll see if we can sneak in a preview of how unmixing and compensation are done in version 11. All right, so let's do it. Theory. All right, so spectral spillover. The thing that we are trying to account for when we are doing compensation on mixing, it refers to simply the idea that we're going to get from every single fluorochrome a range of expression, and it won't be confined to one single detector on our cytometer. So we have to figure out how we can ascribe the correct amount of signal from each fluor, from each detector to the proper fluor. We can do that in a couple of ways. And the first one is compensation, the way that it's been done for a while, going back to FACS scans and FACS calibers and so forth. The general gist here is that if we've got, say, 3 fluorochromes and each of them have signal in 3 different detectors, we can set up an equation where the measured signal, the detector values, are the combination of all the fluorochromes and all the detectors. And what we want to actually solve for is how much actual fluorophore abundance there was. And so you can solve this equation by inverting the matrix, the mixture of what percentage of each of the signals went into each one of the detectors. Okay, so that's just standard compensation. And the biggest aspect of that to differentiate it from everything that's coming next is just that you have a specific detector that's associated with each one of your fluorochromes. You have, fingers crossed, designed your experiment well, designed your panel well to match the tool that you're measuring the data with, and you have a detector in the appropriate location where you get, one of these near one of these peaks from each of the different fluors that you're using. So that's compensation. Those values, the signal that you're using to ascertain how a particular color distributes across all the detectors, come from individual single stained compensation controls. So you'll run a tube, as the graphic here is showing, with just one color in it. And then you'll know that for that tube, whatever the distribution of signal is across all the detectors, that is what a particular color or a particular fluor looks like. So red, for example, distributes a lot in one detector and a little in the others, and so forth. You can use one single stain control for each one of your colors to get the full picture of your system. Okay, so spectral and mixing. Picture on the left is just a repeat of traditional for comparison versus the spectral unmixing case. And in that case, we will just essentially flood the system with lots and lots of detectors. And we'll have many more detectors than we will have fluorochromes generally. And figuring out an exact detector to be the primary detector isn't so important. You can imagine that a number of detectors would do a pretty good job for the blue or the green or the orange color. And we end up basically, though, with the exact same situation where we've got detector values, the amount of fluorescence we measure in a detector, and we've got the distribution of values across each of the different detectors for each of the floors. And what we're trying to do is solve for the actual fluorochrome abundance. The inversion isn't quite so easy because it's no longer a square matrix. For linear algebra fans, a square matrix inverts easily. You can just write an m-1. For a rectangular matrix, you have to do a pseudo inverse, which is what's listed right here, but we can still solve it and get out the fluorochrome abundance. And that's it. So spectral and traditional, very similar. You'll get more measures out of spectral. You'll probably collect more light out of spectral. It'll give you a little more flexibility with your panel design, but the math behind the two is pretty similar. Okay. And so overall, what we're trying to do, what the impact of doing unmixing or compensation is that if you ran a single stain color, a single stain control of some kind, so this is maybe just has just FITSI in this tube, and because some of the FITSI signal appears in another detector that we are taking some part of and ascribing to PE, it looks like perhaps we've got double positive cells, where in fact, if we do compensation or unmixing, we end up realizing that those cells are truly only single positives. That's the goal here, to make it easy for you to visualize data in appropriate proportions to what it's actually expressing. Okay. And then I just list, if you're setting up these controls, what Mario Roeder, my mentor for compensation, what his 3 rules for compensation were. So you have to have controls that are at least as bright or brighter than the samples that you're using. And so you want to show a really bright signal, again, as bright or brighter than the actual data, to see what the amount of fluorescence you're going to, how it's going to spread into detectors, because you'll get more spread with brighter signal. More intensity, more floors, more fluorescence, and so a bigger spread, you'll get the most accurate view if your example, if your control is at least as bright as your data. Okay, background fluorescence should be the same for the positive and negative controls. So this is if you're doing traditional gate-based cytometry. We'll talk about the difference between them. We're just been talking about the positive and negative control. And you're going to want to make sure that autofluorescence is contributing equally to both the example of cells that bound your fluor and cells that didn't, so that if you're using that as the metric for is this properly compensated, differences in autofluorescence aren't contributing different values and causing poor compensation. Comp controls must match the exact experimental fluorochrome. So this is the controls for the floors. So the antibody doesn't have to be the same, but the fluor does. So it's not proper to substitute FITSI for GFP or something like that. You'll just get slightly different signals and then poor compensation. All right. So that's the basics behind there. And then the actual math for the compensation methods that you can put in play. You've got 2 choices, the detector arrangement and the optimization target. And each one of those choices has 2 options, and you can mix and match between them which one you want to use. So we just covered in some detail the difference between traditional and spectral compensation. So big difference, 1-to-1 parameter to detector ratio and 1-to-many parameter to detector ratio for traditional versus spectral. And then for what am I trying to accomplish? What's my optimization target that allows me to know that this compensation or unmixing is well done? Well, you can either do gate-based, which is a more traditional approach, where you'll have a negative population and a positive population on a single stain control. And for the whatever fluor it is you've added, they'll be separated, positive and negative. But for every other fluor, if this is a single stain control, you should have the same signal between background and positive. And so we can check the MFI on every other secondary channel and make sure that it's equal between background and positive population. So for that, it's called gate-based because we use gates to identify a negative and a positive population. Alternately, you can use something called AutoSpill, which is relatively new. It's a few years old now. We worked with the Babraham Institute to help bring this to life. What we do is do a regression line between those negative and positive populations, except we'll use, instead of negative and positive populations, we'll use the whole data within a cleanup gate. make a regression line, and try to make sure the slope is flat, which would imply the same thing as equal MFIs. And I will show you a couple of pictures to make this more clear. And then just again to emphasize that one is an optimization target here. On the right, the choices between gate-based and AutoSpill, and the other is the detector arrangement, and you can mix and match. So I could use traditional and AutoSpill, spectral and AutoSpill, spectral and gate-based, or traditional and gate-based. Any of those are going to be fine. Okay, so this is the AutoSpill paper. Principled framework that amplifies the analysis of multichromatic flow cytometry data. And that is, okay, there we go. There's our Carlos Roca as the primary author. Nice work by him. So the gist here, linear regression to calculate the exact same kind of coefficient matrix that we were solving for when we were solving the math problems there. And AutoSpill is going to work in an iterative manner. So what it'll effectively do is figure out the whatever the compensation or unmixing matrix is for a given data set, apply that matrix to the data, and then recheck the goodness of, do I actually have flat regression lines? And if not, we can treat that unmixed or compensated data as the new input data set and modify it a little bit until we get the best possible compensation or unmixing matrix that we can. This tool is also useful for subtracting out autofluorescence, in that we can just fit autofluorescence like another parameter that gets a linear regression line. So I think the overall effect of AutoSpill is what I have in blue there, easier cytometry. So I'm a big fan. I like AutoSpill a lot. And I will show you a little example to illustrate how it works. Okay, so no matter what combination of methods you're using, you start with a cleanup gate. Cells here using forward scatter and side scatter to find things that are roughly the same size and roughly the same complexity, so that we can assume the autofluorescence contribution of all those cells is relatively equal. If you're doing gate-based compensation on mixing, from inside the cleanup gate, we make two gates, a negative and a positive. I've shown them here. And what we'd like to do is say, if this is the single stain control for whatever's in R780, then for R820, those signals should be the same. They're not at the moment. So that is the spillover. And we will use our mixing or our compensation matrix to figure out what that spillover is, subtract it so that we end up with MFIs of these two populations being equal, and then we'll know we've done a pretty good job with the compensation or the unmixing. That applies that your comp controls have followed the three rules that we pointed out earlier, and yes, they are appropriate for your actual data. But we know if the MFIs line up here, then your comp controls will at least properly compensate or unmix. Okay, if it is AutoSpill instead of gate-based, we fit the regression line through all of the data that were in the cleanup gate. And then we use our matrix to try to flatten that line and know that we've done good compensation or unmixing with a slope of 0. And the inversion is as normal. So there is nothing different you have to do in terms of setting up your experiment if you're going to use AutoSpill. Single stain comp controls, following the three rules, feed it into FlowJo, and you can do it. Okay, and I've called it robust linear regression a couple times, so just to explain that, there's a weighting process. Any events that are farther away from the regression line, we just weight them less. So a few outlier events cannot have an outward or untoward effect on your matrix. Okay, and then one benefit, or no, one, I think, important benefit of using AutoSpill is that if you have any controls that look like this, and this is very, very, very possible, anytime you get into higher dimensional panels, I think it's very likely that you'll have, say, a lot of negative cells for at least one of your colors, and then a decent number of positive cells, but much less than the negative population. And maybe there's, even if you're doing cells instead of beads, there'll be a range of expression levels. So gate-based makes it hard to automatically identify that positive population, but for AutoSpill, since we're just doing a regression line through all this data, no problem. So it'll make it a little easier if you've got a handful of controls that aren't wonderful. Okay, so how do we do it in FlowJo? So this is just showing the compensation wizard, and I've used the blue boxes to highlight the particular aspect of the tool that I'm showing about. So to do traditional compensation, you will leave the traditional box checked off right here. This is gate-based. That's the traditional manner here. There is a checkbox for spectral that is left off. So this is gate-based and traditional as opposed to spectral. As soon as you load in all the floors, they're matched one floor, one parameter. And then we go about the process of trying to automatically gate the cleanup gate and automatically gate the positive and the negative population. You can go in and hand edit anything that the automated algorithm does not do particularly well, and then do your compensation or your unmixing. Okay, if you're going to do AutoSpill, the only difference is you will then check the AutoSpill box right here, and then you'll notice the positive and negative gates go away because we don't use them. All we're using is the cleanup gate. And then it proceeds as normal. If you're doing spectral unmixing, you can use either gate-based or AutoSpill, and then check the spectral box. That's all it takes to switch to spectral unmixing. You will notice to begin with, you're going to have a whole bunch of parameters. You'll generally select use all the detectors. And so you'll get a whole bunch of detectors that you're not going to create a parameter from. You'll say, okay, I've got one or two visible on the screen, and we'll make parameters out of them. And you'll still either, again, use gates if it's gate-based, AutoSpill if it's not, to identify whatever your target is and try to maximize it. And then you will want to you can right-click on any one of these empty boxes, and there's a choice for remove all of the unused detectors. The idea there is that we're only going to create a parameter for as many floors as you have. So even if there's 50 detectors, and if you only load in 4 floors, you should only get 4 parameters. And so it's not removing the detectors from the calculation, it's just removing them from the list of how many should I make a parameter for. Okay, so it's essentially saying something like, the primary detector is fairly unimportant, but we're going to accumulate all the signal that we have ascribed to FITSI and call it the FITSI parameter. Okay, if you're doing autofluorescence extraction, there's 2 ways to do it. If you choose to use AutoSpill, you'll use one tube that is unstained, and that's really all you have to do. Make a cleanup gate, and AutoSpill will fit a best fit regression line to it. Whatever the variance from flat is, we assume that is the contribution of autofluorescence. If you are doing a gate-based approach, there is an option within the negatives to pick true zero. So you pick for some unstained cells, whatever population is visible as your positive, and then true zero is the negative, and we assume whatever whatever fluorescence there is the contribution of autofluorescence, which is a fairly good assumption, a little simplification, but it works well enough. You can have multiple different cell types, create multiple autofluorescence parameters if you want to break it down a little bit further. And on this slide, it'll just mention, if you're going to try to do autofluorescence extraction, you'll want to try to match the cell type. If you've got a lot of different ones, maybe you'll do multiple channels for it. So a nice choice is to assign autofluorescence to an empty channel with the highest signal level. And so that's just, I'm just showing the spectral population plot here in FlowJo, where you might take your unstained cells and then glance at which one of the detectors it has the largest signal in as a way to figure that out pretty quickly. Okay. All right. You can get a couple of metrics to assess your compensation or unmixing. One of them is the spillover spread. We'll calculate a spillover spread matrix, the SSM. So the unmixing matrix or the compensation matrix itself will only say something like, compared to the primary detector, some other detector had 3% of the signal. Even if that number goes up as large as 100, it just tells you you had 2 detectors that were pretty close and you got equal amounts of signal in both of them. So it doesn't necessarily mean anything's wrong with that. It means maybe some better panel design could have been used to create less need to do large unmixing, but that's okay. We can do it. However, spillover spread, what that's going to do is tell you once you do your compensation mixing, how much more has the positive population spread compared to the background population. And so that is a real number that we can look at and say, okay, so this is well done. This particular panel is pretty good. The compensation worked well. General rules of thumb here, if you can have numbers under 5, so for a particular color in a particular detector, that's really nice. The range of more than 5 up to 10 is, decreasingly usable. And great if you hit double digits, that's probably a combination within your panel that you don't want to actually have to use. So if you've got a large experiment, it doesn't mean that you have to make every combination within that experiment, every fluor and every detector, something, something super useful. If you want to make sure that your critical parameters have very nice SSMs, you can define a panel in such a way that if you've got a few bad spots, you put things that you might not look at together in that combination. Okay. The SSM is normalized or scaled to the staining index. So a very, very bright single positive that has a decent amount of spreading to it might give you a very small SSM number. The assumption there is that you're willing to accept a little more spreading to get such nice separation between background and single positive. The TSM is another option under the SSM tool in FlowJo that will give you the same value but without the normalization. So it's just telling you what the total spreading is, regardless of how bright the signal is. So we've found that maybe looking at the combination of those two numbers can be helpful. That where you notice a very large number in TSM but a small number in SSM, you'll know that you're going to add, you're going to add some spread, even if you might have a nice resolvable signal for that particular color. Okay, we also will do spectral similarity. So that is going to tell you if two particular colors are similar in terms of their pattern of expression across all the detectors. So a 1.0 in this metric, and we're showing it here in the compensation wizard. You can also get it out of that spectral population viewer. 1.0 means it's the identical fluor. So you've put two things that are exactly the same essentially in there. Rule of thumb for that one. And so this is just a heat map view where orange is something compared to itself. So that's the perfect 1.0. And then you'll be doing better work. The more whites and light yellows you use, the closer you get to the dark orange, the more two dyes are going to look the same and have some difficulty being unmixed. Rule of thumb is, I've heard people say like 0.93 is even okay. If you can keep it to 0.9, even better. And like lower is just better, basically. All right. So that is spectral similarity. And that is the end of the slides. So fingers crossed. That was why do we do compensation? What are your choices? How do you do it? What are the buttons in FlowJo to hit? And now let's actually do some in FlowJo. All right. Oop. I have a version of this webinar already done, just in case. But let us start from scratch. There's still Zoom controls up. Wonderful. Only took me 18 minutes to get past that screen. Okay, and the chat is still empty. Looks like we are doing okay with people and questions. So I'm going to bring in some data from the A5SE. We've got some comp controls, and I've got some full stained samples as well. Okay, and of course, we will work on the comp controls. So 28 color data. So I'm not sandbagging you here. We're using some very, very realistic experiment where the high parameter land. One trick I'll mention, if you have a particular cleanup gate you think is going to be appropriate for your data, you can actually just draw it and apply it to the rest of the data. So I think I generally want to just grab that big lymphocyte population, call it whatever you want, but I will call it cleanup, and copy paste it to the group. The algorithm in FlowJo will automatically create a gate if you have not done this. Sometimes it creates sort of an odd shaped gate, a little smaller, a little bigger than you might like or something like that. So up to you, unimportant there. Okay, so I'll mention these are groups at the top of the FlowJo workspace. The ones that have the word comp or unstained automatically go into this compensation group. If you click on a group that has the raw compensation, you'll notice the comp wizard is colored and ready to go. If you click on any other group, it is grayed out. If you have less than two samples, if you've got one sample or zero samples, there's nothing to compensate or unmix, so it will also be grayed out, but we're ready to go here. If your comp controls are not named with the word comp or unstained, you can either just drag and drop them into the group, or you could go in and modify your preferences. So the heart is the preferences in FlowJo, compensation is here, and figure out what pattern is you name. If you use like the word single in your single stain controls, you could say something like add $ file name contains single. And that we will automatically identify them as single stain controls and put them into a compensation group for you so that it happens automatically, because automatic is nice. All right, files in group, hit the wizard. Because this is a spectral experiment, we've got more detectors than we have parameters that we're going to create. FlowJo up front doesn't know that you're going to choose spectral, so it says, would you like to select the detectors to use just in case? This is traditional, and you want to match exactly the number of detectors to the number of floors. But in almost all cases, you can just select basically everything, say choose select parameters, and figure it out later within the wizard exactly what you want to use. Okay, it'll automatically match up which control it thinks is the exemplar to create each parameter with. Okay, so big list, 28 or so. And at the top, we have the assignments of which control, comp control, CD57 FITSI B510, assigned to the detector B510 and the CD57 FITSI parameter. This bit of text that has the parameter names, somebody who collected this data has annotated it for me. It doesn't have to be annotated ahead of time. You could type it in here afterwards. At some point, you've got to name the parameters. This is nice that it's already in here. And so what I'm going to do in this case will be to actually make sure that the same primary detector was used for each one of my controls to make sure that I save me a little bit of trouble of not having to rename them. If we assume that the person who designed the panel did an okay job, then good enough there. So I'm just going to scan down the list where it's green. FlowJo has found a positive and a negative peak and also has the name of the parameter in the name of the actual control sample. And so green says, I think we got this one right. Like we have two things that confirm this is it. If it's red, maybe we didn't use that particular detector to make a parameter out of. We can remove those later. If it's yellow, one of those two things, oh, hey, save my workspace. Why not? Let's call it the webinar. Okay, one of those two things is true. Couldn't find a particularly good peak. The name wasn't in the sample. Doesn't mean there's something wrong. A lot of times you'll name your comp control something that doesn't have the name of the detector or the parameter in it. It's okay. It doesn't mean it's wrong. Anyway, so glancing down, it's all looking pretty good all the way through here. I'll keep scrolling. Everything's looking pretty good. There's our autofluorescence control. Okay, here's one where we have a different parameter assigned to the different detector. I'm just going to remove this detector and I will go back and put the control on YG780. So here's a list of all the different comp controls. So I could put a different comp control onto that particular detector. I also have just to remove this parameter, remove all the unused. I will remove this parameter for the time being. Okay, still looking good, looking good, looking good, looking good. Here's one. There we go. I would like to add the control from BV480 to this detector. It was probably assigned elsewhere. Look, it was assigned to the one next to it. If you have enough detectors and it's off by one, it probably doesn't matter whatsoever, but just so I don't have to rename them, I'll go to that trouble and then I'll give this one back BV510. Or something close to that. That was probably there. Okay. And 605. That was the one we found earlier. Copy that one. Okay, that was somewhere else. Oh, no. That is down below. It's this one here. Oh, that's IGD. Where did IGG go? And anyway, to sort, I can just do, no, I guess I do not have that one in this panel, so I will remove that one then. And I can type RE to remove a given parameter. Okay. And so here's just another case of like the controls are shifted by one in a couple of places compared to what the original panel designer wanted to do. V510 seems to have CD122, but you selected CD25. Oops. Good call. What is that under V510? Where is that one? All right, well, we maybe don't have that one in this panel either. Let's remove that one then. Ran a couple of iterations of the panel, so not every single one of the samples and detectors was used, and I just kind of left the names from the past. B78. It's probably might not have that one. All right, that one's okay. That one is not. So anyway, probably the most time-consuming part of this thing is just making sure that the names are in properly at some point. YG670 is right there. Oops, except I want a YG670 here. And I want a YG660 here. And the 602 or 585. 585 there. All right, and that was 7AD. So a couple shifted left and right, one detector, but we'll get the names right. All right, I think I got them all now. Other than just make sure that there isn't a 123 in here. So that was 13. No, okay, so there's no 123 in this. All right, so now that I've got those lined up, I will right click on one of these and say remove unused parameters and clear that list out down to just the things we have. Okay, so a couple of red circles left here, which implies we're missing either a positive or negative gate since we're still in gate-based compensation. We can see where there's a few blank spots. And so time to go look at the data and fill in anything that's missing. Okay, so scrolling down, we're okay. Here's one where sometimes the gating algorithm just misses one. So I'm just going to double click on the plot. I'm going to draw in the gate that's missing. And then from the list here, I will say what gate I want to be my negative example. So this part I'll have to do if I'm doing gate based. If I do AutoSpill, which I'll do next, I don't have to worry about it. All right, all that seems okay. There's one again where for whatever reason, it just missed the really big, obvious bright peak. So I have to tell it what the negative population is. You'll notice I'm not working super hard to get a perfect gate here. The idea is that for however big your gate is, what the program's going to do is calculate the median fluorescent intensity, which is shown as the dotted red line. So moving the gate a little right, a little left barely moves the red line. So it doesn't make a huge difference there. Exactly how carefully you make that gate. Let's see. And I'm just searching for things since I'm calling them all neg, just to say neg to fill it in as please use this as the negative example. Okay, this one is our autofluorescence. And so I have to put in, if I'm going to use autospill, then it's okay. If I'm not, then I have to put in the true zero choice as my example of negative. All right, and one more here to move it. Get this right. G. Yep. It's like I've got 4 more to take a look at. It all looks like it's pretty much the same thing where we had a nice, beautiful peak that for whatever reason, the algorithm didn't identify as a peak. Let's adjust that one a little bit. That one, that might have mattered, that was so far towards the positive end with the negative gate. Dun, dun, dun. Okay, here's a good example. There's no apparent positive peak in this one, so we might wonder, what should we do? This is probably one of the few cases where my apply to all cleanup gate was not a good choice. We can see this is associated with CD14, so we'll guess the cells with CD14 are actually up here. All right, there we go. And now we can AutoGate that. Boom, done. I might adjust this one too. This is, so this is live dead. Live dead, always a funky one because almost all the rest of the samples, your algorithm will be looking for, let's see where there might be a bunch of dead cells. Cool. Your algorithm will be looking for live cells, happy, healthy live cells. Live dead, you want to make sure there's some dead cells in it. So I'll throw it down there where the junk is. Okay. You will notice that when you've got a positive and negative gate if you're doing gate-based for every one of these and every one of the samples with a control, it'll say finalized in the upper right-hand corner. And so then at that point, we can view the matrix. Okay. Matrix preview window pops up. The blue is our uncompensated. I can turn that off if I'm pleased with that. We're previewing it on one particular compensation control, but we could also preview it on a full stain sample. As the comp control should comp okay, the full stain ones will be more interesting. A little bit of background angle to that stuff, some co-expression. The other thing you might want to do is I'm going to go back into my full stains, and I'm just going to draw some kind of minimal cleanup gate, or maybe a couple of cleanup gates, and then I can grab single cells, so that we can preview the events on decent data. And let's just do, sure, we'll just do that and grab the bulk of the cells here and get the things off the axis. Apply those gates to my group, so it doesn't matter which one of the full stains I looked at. And then from the preview population button, I have the ability to clean up this data to just look at the good stuff and not judge my matrix based on some of the worst things. Okay, and we might want to adjust our bi-exponential for some of these as well. Do a little extra width basis on BB660. If you want to do anything like that, it's just a matter of double clicking on one of the samples. Yeah, a little wider. And the T for customize, and you can, if you want, add a little extra width basis to compress it off the axis some. And see if that tightens up the negative region on BB660. All right, a little bit. Okay, so that is doing, and let's minimize this, that is doing traditional gate-based and non-spectral. So let's switch it up and do AutoSpill. I click the AutoSpill button, and all of a sudden, we see all the gates have disappeared. The matrix has a new name. We could go back and rename these things at some point to tell them what they actually are. I don't care at the moment. I just call it matrix -1. Let's make this not that blue color, since that is the color that gets used for the uncomped, and I'll say view. So AutoSpill will do an iterative approach, so it'll take a little bit longer. But again, you could have saved all the trouble of making all the gates, which, as you all had to sit and watch me do it, you can know that it took a little bit of time. So save some of that gate adjusting time and just run the algorithm, which will again do an iterative bit where it tries to make the best possible compensation matrix or unmixing matrix, depending if you've checked the spectral box or not. All right. How are we doing? 7:58. So I am running behind, and this is taking a little while. I wanted to very much show you V11. I might just cancel for now, mention that you can hit spectral, either one of these things, hit the view matrix button. It does the same thing. you know, Spectral's now doing spectral calculations using AutoSpill, and just pop open. I had a completed version of this workspace. Done over here. I'm going to double click on spinning color wheel of death. Awesome. Just to give you a view of the completed matrices. Oh, don't take forever right now. We only have 2 minutes left in this webinar. I still want it to break out V11. All right, I am going to force quit. It's a good day when you show up late and have to force quit. And open up the saved comp webinar workspace, just to point out that each time that you run one of those, it makes a new matrix, and then you can compare them. So this should be brief, and then I'll run 5 minutes over, and I will give you a preview of how the same panel looks in V11. All right, double clicking here. If you run all four, and I bothered to name them as trad gates, traditional autospill, spectral with gates. You can turn off the uncompensated again. The gist of this is that if you want to assess how good they were, you can look at the SSM, which is right here. Again, it's highlighted, so light pink numbers are nice. If it gets up over 5, you start getting darker numbers. This is a pretty good effort. I will assume that the other one, which looked a little funkier, maybe I missed a detector assignment or so. Anyway, so that's the SSM. The TSM is right there. Don't have that one heat mapped. Version 11 will. If we want to compare two different approaches, you can highlight them both. And so I've highlighted the orange and the green, the spectral with autospill and the spectral with gates. And you'll see we plot the particular cells for each one of these. So you can see which is which. We can preview the population and do, like I did, cleanup gate and a live gate to get rid of some of the junk from the axis. And you can see there's not a whole lot of difference between them. Yeah, it looks like actually they're essentially identical. And the numbers up top become the delta between them. So generally really small numbers. The only place we see a bit of a difference is in the autofluorescence, where it was done two different methods, where AutoSpill does the line and the gate-based uses the true zero. Okay, we're going to go a couple more minutes here. That is the V10 version. I just want to show you the V11 version. That is coming in the end of May or so. Okay, so V11, immersive, has dark mode, etc. I'm going to hit new workbench. And the only thing we're going to look at today is compensation, which is this context right here. I guess we can leave the graphics up. Let me just grab in the same data. And why not throw the full stand in as well? And we won't go through every single bit of this, but I'll just show you how this is working. The gist of 11 is that at the top, the grouping is a little more differentiated at the beginning and also is going to be hierarchical. And so it's already figured out that amongst all of the data, all the acquired data, some is comp, some is experimental. And here's particularly the comp controls for this A5SC experiment. And then if I say, take me to the wizard, we can get started. It's using the hierarchy as the selection tool for everything. And it is going to show you a spectral plot right off the bat as we move through the data. It has a list of the samples down here. We can expand that all the way up if we want, or compress it to see our plots. Mousing through, we just use this feature wheel right here to show each of the selected samples as we can move through. Here's your same choices for traditional or spectral, and then AutoSpill is on or off. It is on at the moment, so it's not showing any gates on these data files. Down below, let me expand this again a little bit. It's just showing you what it's done. It is AutoSpill, so there's no gates down here, but it's just showing you you've got the samples. Are we using them? Is it a color control versus is it an unstained control? Is what the colored wheel means. Mousing over everything tells you what it is. and which detector this was assigned to. So if there's any that were assigned to the wrong detector, and just scrolling down, 660, 710, 675, 730, 750, 450, I think they might all be right. Uh, I was assuming I'd have to change at least one, since there were so many we had to change in the other in FlowJo 10. Um, might all be right. Dun, 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 dun. All right, well, if I wanted to change one, I'd hit the drop down, and then it's just the list of all the different detectors that you can pick from, or you can type right into the box to search. Okay, so that went pretty well. All right, if you are pleased with your detector assignments and you're using AutoSpill, you can just say preview this matrix. This will think for a second, and then it's going to pop out a preview, and you can use the hierarchy to control exactly what it is that you are looking for on the data. Hmm. There we go. All right. Spillover matrix up here. And then you can just click to the buttons if you want the spillover spread, if you want the similarity, turn heat maps on or off, pick which particular file that it is that you are looking at. We've given up the idea that we can show everything all at once. So we just pick a particular parameter, give you a plot type you can choose from. So maybe like contour, turn off the uncomped if you don't want them, show the other detectors if it's spectral and you want to see things that are fluorescing in other channels that you might not expect. And if you wanted to actually pick a different population to visualize, you just click it out of the list there. So different parameters, holding firm up there. If you want to pick a different sample to look at, you just pick it out of the list over here. So V11 a lot different. We're just using the hierarchy for most all selection tools and so forth. At the point you like the matrix, say create. And a matrix will appear, and we have a separate group of compensation matrices over here. You can then drag and drop those to any particular file you want. And so that is the 5-minute version of how you do compensation or unmixing in version 11. So just a teaser. Again, this is slated for release at the end of May. So we have 3 more months to polish this thing up. Okay, that is all I'm going to do for tonight. We can, I guess, drag a matrix for the fun of it, drop it on a sample. You can drop it on a group as well. You can see that the colors persist, but that is all exactly how FlowJo 10 has always worked. So that should be no surprise right there. All right, the chat looks like it is clear. And let me just make sure there's nothing in the Q&A box. All right, that looks clear as well. So there is a question that just popped up. Can FlowJo unmix data from Cytek cytometers? Yes. You just have to have the single stain controls. And so putting them into the, choosing spectral will be fine. There's a couple questions of, will this recording be made available and can I get the recording? The answer is yes. Every one of our webinars get recorded, and dun, 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 go on the Learn tab here. This is our new updated website. So upcoming webinars are here. Here's the one we're doing right now. And then all previously recorded, this one will pop up in a couple of days. And so you've got all the previous ones we've done, as well as wherever it went, there it is, the way to search and filter for particular things that you're looking for. Okay, well, I appreciate that someone asked for the recording, and that must have done something okay, as opposed to crashing FlowJo and starting late. Let's see, we've got 2 more open. Can FlowJo unmix Sony spectral data? The answer is generally yes. We have run into some Sony data where we do a poor job of figuring out what kind of FCS file it is. Is 3.0, 3.1, 3.2, etc. And it gives us some problems. Still working to improve that, but I think in general the answer is yes. And then specific cytometers, their newest cytometer, I think puts out is it a 3.2 version file? Anyway, I've heard some folks have trouble with Sony. So good question, Francis. All right. Any other questions? There's a question, does the Sony ID 7000 need compensation? I believe that is a spectral machine, so it would use unmixing. Popular day for Sony questions. Okay, let's wrap for the night. Yes, it has unmixing option. Cool. If you have further questions, so unmixing is what you would do instead of compensation. Compensation just means that you would do one detector per one parameter. Whereas if you're on a big fancy, um, spectral cytometer, then you'll use unmixing and it'll do a nicer job. Okay. My email address is, I'm just going to type it in again, john.flowjo@bd.com. I can be reached for questions whenever. Thank you for joining me. Apologies again for the late start. Have a great, well, for me, it's evening. For everybody else here, maybe it's a different, you're in a different time zone. Maybe it's day. Have a good one. Take care.